Now I am going to start third unit of Kingdom Biological Classification. This is very important unit. On an average, 1 or 2 questions is asked from this unit. At least one is asked from this unit. Okay, and that is Kingdom Fungi. So this Kingdom Fungi includes heterotrophic organism, which are multicellular with loose tissue organization. So this is a unique kingdom of heterotrophic organism which are multicellular with loose tissue organization, except yeast, which is unicellular. Now when you look into the habitat and morphology of fungi, they show great diversity in terms of habitat and morphology. They show great diversity in morphology and habitat. Okay, now so where you can find fungi. Fungi can grow on rotten bread, then on fruit, then mushroom on dung, then parasitic fungus. So there are various kinds of fungi. Okay, and there are various economic importance of fungi that we will discuss later on, or as it will come. Now they are cosmopolitan. That means can occur in air, water, soil, and on animal and in plant. Now then where they do prefer to grow? They prefer to grow in warm and humid places. So preferred condition is warm and humid. Okay, now then with the exception of yeast, which is unicellular, all fungi are filamentous. Fungi are filamentous, and their body, that is vegetative part of that fungi, is consist of hyphae. Okay, which is a cylinder thread-like structure. Okay, now this hyphae forms a network, and this network of hyphae is referred as mycelium. Now when you observe to hyphae, what you will observe, hyphae is something like this. Now this hyphae can be of two different types. First, without cross wall, multinucleated hyphae, or with cross wall. So this is referred as aseptate hyphae or multinucleated hyphae, or with cross wall, that means septate hyphae. So some hyphae are continuous tube filled with multinucleated cytoplasm. These are called cenocytic hyphae, that is multinucleated hyphae or aseptate hyphae. Other have septate or cross wall in their hyphae. Now when we talk about cell wall of fungi, the cell wall is mainly made up of chitin, but in one case, that is in case of members of Oomycetes like Phytophthora, Albugo, Pythium. Okay, in these cases cell wall is made up of polysaccharide cellulose. Okay, so in some cases cell wall is cellulosic. Okay, now most of the fungi are heterotrophic and absorb soluble organic matter from dead substrate, and hence are called saprophytes. Those that depend on living plant and animal are called parasite. Then some are symbiont, like in case of lichen and mycorrhiza. Okay, now when we talk about reproduction in fungi, it can be vegetative, asexual, or sexual. So vegetative means by fragmentation, fission, or budding. Asexual reproduction by spore called conidia or sporangiospore or zoospore. This zoospore is motile, whereas conidia and sporangiospore are non-motile. Okay, now sexual reproduction by oospore, ascospore, and basidiospore. Okay, so these three types of sexual spore, that is oospore, ascospore, and basidiospores, are produced. And these spores are produced in a specialized structure which is referred as fruiting body. Okay, now when we talk about sexual cycle of fungi, okay, so sexual cycle of fungi is divided into three different stages: plasmogamy, karyogamy, and meiosis. So first is plasmogamy, where fusion of protoplasm between two motile or non-motile gamete called plasmogamy. Okay, then second is fusion of two nuclei called karyogamy, and third is meiosis, that is reduction division. Okay, third is meiosis, that is reduction division, which results in formation of haploid spore. Okay, now so at the time of sexual reproduction, what you will observe, this is very important. Okay, now So at the time of sexual reproduction, what you will observe, there are three steps: plasmogamy, karyogamy and meiosis that I have discussed. Plasmogamy, karyogamy and meiosis. Now, in case of lower fungi, okay, when there is plasmogamy, suppose there are two cells, suppose this is one cell, this is second cell, N. There is plasmogamy and that will result in formation of dikaryon, N plus N, that is plasmogamy. In case of lower fungi, plasmogamy is immediately followed by karyogamy that results in formation of diploid zygote, and that will immediately undergo meiosis to form haploid spore. Whereas in case of higher fungi like Ascomycetes and Basidiomycetes, this plasmogamy is not immediately followed by karyogamy. So there is an intervening phase referred as dikaryiophase, and that is of variable duration, and after that karyogamy and meiosis will take place. So let's see. Two haploid hyphae of a compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell. However, in other fungi like Ascomycetes and Basidiomycetes, that is higher fungi, an intervening dikaryotic phase, that is N plus N, 2 nuclei per cell occurs. Such a condition is called dikaryon and phase is called dikaryiophase, and that is of variable duration. It is not a fixed duration. Later on, the parental nuclei fuse and the cell become diploid and the fungi form fruiting body in which reduction division takes place. So reduction division always takes place in fruiting body. That is reproductive structure. Okay, and leading to formation of haploid spore. Now, when we talk about classification of fungi is divided into different classes on the basis of following criteria. Okay, first is morphology of mycelium, that is whether it is septate or aseptate. Second is mode of spore production, whether it is exogenous, that is within the body, or outside the body. Exogenous means outside the body, endogenous means within the body. Then fruiting body, what kind of sexual structures or reproductive structures are produced. So these criteria, that is morphology of mycelium, mode of spore formation and fruiting body, are three criteria on the basis of which fungi is divided into different classes. So we have to study four different classes, that is Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. Okay. So first is Phycomycetes. Okay. Now, Phycomycetes or lower fungi, they are found in aquatic habitat and on decaying wood, in moist and damp places, or as obligate parasite on plants. Okay. Here mycelium is aseptate, that is without cross wall, and synocytic, that is multinucleated. Asexual reproduction takes place by zoospore, that is motile spore, or aplanospore, that is non-motile spore. Now these spores are endogenously produced. Endogenously means within a fruiting body. Then a zygospore is formed by fusion of two gamete. Okay, so when there is sexual reproduction in Zygomycetes like Rhizopus and Mucor, there is formation of zygospore. These gametes are similar in morphology, we call it isogamous, or dissimilar, we call it anisogamous or oogamous. Okay. Now, some common example are Mucor, Rhizopus, Albugo. So here name is not mentioned, but you have to remember two names. Most important name, Albugo, which is responsible for white rust. Okay. Second, Phytophthora. Okay, this is responsible for late blight of potato. You have to remember these two names. These are very important example. Invariably question was asked from this part. Okay. Now next is Ascomycetes. Asco, okay, commonly referred as sac fungi. It is commonly referred as sac fungi. Okay. Mostly multicellular, example penicillium, or rarely unicellular like yeast. Okay. Yeast belongs to this, that is Saccharomyces. They can be saprophytic, decomposer, parasitic or coprophilous, growing on dung. Okay. Now mycelium is branched and septate. We have septate. Now this is important point. Asexual spore is called conidia, produced exogenously. So conidia is asexual spore in case of Ascomycetes, which is produced exogenously. Now on the special mycelium called conidiophore. Okay. Now conidia on germination will produce mycelium. Sexual spore are called ascospore, which are produced endogenously in sac-like structure called asci or ascus. Now these asci or ascus are arranged in different manner in a fruiting body, and that fruiting body is referred as ascocarps. So fruiting body of Ascomycetes is referred as ascocarp. Now some example are Aspergillus. Okay, Aspergillus niger is source of citric acid. Then Claviceps. Claviceps is responsible for a disease ergot of rye. Okay. Then Neurospora. Neurospora is used extensively in biochemical and genetic work. Okay. Now then many members like morels and truffles are edible. Okay. Morels and truffles are edible and they are considered as delicacy. Now this penicillium is used for production of antibiotic. Penicillium notatum. Then yeast, Saccharomyces, is used for production of bread and beer. Okay, baker's yeast and brewer's yeast. Okay, so we are through with Ascomycetes. All those information which is present in NCERT. I am confining myself only to NCERT. Okay. Now, next, Basidiomycetes. Now, Basidiomycetes is commonly referred as club fungi, and this is most advanced group of fungi. Okay. So, members are most commonly seen, okay, in nature. So, members are mushroom, bracket fungi, puffballs. They grow in soil, on logs and tree stumps, and in living plant body as parasite like rust and smut. Rust is by Puccinia and smut by Astilago. Okay. Now their mycelium is branched and septate. The asexual spore are generally not found. Very important point. In Basidiomycetes, there is no specialized asexual spore, and also sex organs are absent. This is very important evolutionary trend. When you move from lower to higher fungi, what you will observe, sexual structure gets simplified. In Basidiomycetes, you will not find any sex organ. Okay, but vegetative reproduction by fragmentation is common. The sex organ are absent. So in Basidiomycetes, there is no sex organ. But plasmogamy is brought about by fusion of two vegetative or somatic cell of different strain or genotype. That means one is plus, another is minus. One will act as, these are different genotype or different strain. One will act as male, another will act as female. So plasmogamy is brought about by fusion between these cells. The resultant structure is dikaryotic, which ultimately give rise to basidium. So basidium, where karyogamy and meiosis takes place in the basidium, producing 4 basidiospore. Now, basidiospores are produced exogenously, whereas ascospores are produced endogenously. This is one of the difference between basidiospore and ascospore. Ascospore endogenously, basidiospore exogenously. Okay, on the basidium or basidia. The basidia are arranged in fruiting body called basidiocarp. So fruiting body is called basidiocarp. Common example are agaricus, which is edible, Astilago, that is smut fungi, Puccinia, which is rust fungi. Okay, so we are through with Basidiomycetes. Now last group is Deuteromycetes. Okay, now Deuteromycetes is also referred as fungi imperfecti. Fungi imperfecti. Okay, or imperfect fungi. Why we call it imperfecti? Because only asexual or vegetative phase of these fungi are known. That is, so far in members of this group, sexual stage is not reported. Okay, only asexual stage is there. Now, when the sexual form of these fungi are discovered, they were moved into class they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. So it is not like that ki this group of this member do not have sexual stage. We are not aware of that sexual stage. And once sexual stage is discovered, fungi is moved out of this group to place where it rightly belongs, either Ascomycetes or Basidiomycetes. Okay. Now, later when linkage is established, fungi are correctly identified and moved out of Deuteromycetes. Once perfect stage or sexual stage is discovered, they were often moved to Ascomycetes and Basidiomycetes, either into Ascomycetes or Basidiomycetes. The Deuteromycetes reproduce only by asexual spore known as Conidia. So, Conidia is asexual spore also in Ascomycetes, also in Deuteromycetes. So, both these group reproduces asexually by Conidia. Now, okay, the mycelium is septate and branched. Some members are saprophyte or parasite. When large number of them, this question have been asked, a large number of them are decomposer of litter and help in mineral cycling. This question was asked in I think in year 2016. They play very important role in mineral cycling. Some example are Alternaria. Alternaria is responsible for early blight of potato, Colitotrichum. This Colitotrichum is for red rot of sugarcane. Okay, and Trichoderma. Trichoderma can be used as a biocontrol agent that we will study in microbes in human welfare, or it is also Polysporum is also source of Cyclosporin A, which we will study in microbes in human welfare. So these are example of Deuteromycetes. So we are through with kingdom fungi.